So thanks everybody for attending. I realize these are crazy times, as I was saying earlier, and so I know how difficult it is to coordinate and find time, and also for David to spend pretty much the whole day with us from 8 o'clock in the morning till now, and interviewing for this position. I hope you've all had a good time. I had a wonderful virtual breakfast with him, and I'm looking forward to his seminar now. So David comes to us, although electronically, all the way from Umia University, which is a wonderful, highly dynamic powerhouse in ecology and evolution, which I've had the privilege of visiting before. He's an assistant professor there. He got his PhD at the University of Virginia and did his undergraduate at the University of Vermont. He has a number of awards in science and innovation and sustainability. He has close to 50 publications, or something outrageous like that, and his work is in some of the best journals in our field. And his work is really cool because it synthesizes theory and empirical work, which I think is very much the way all of the faculty in E3B approach scientific research. So that's really nifty. And he uses whole ecosystem manipulations, which has proven to be a powerhouse, especially in limnology, but also just in the general field of empirical testing of theory, where you can get a better handle on the scale, even if sometimes the price is for replication. And he's advanced our understanding, I think, or at least the work that a lot of us are most impressed with in understanding state changes, or how a system will flip from one system to another, one state to another. And that's really important in understanding tipping points, and also in trying to develop early warning systems to know when a system is going to change, and perhaps create environmental issues that need to be addressed, especially in this day and age when most ecosystems are being pressured by a number of environmental changes. His work has scaled up globally, and because he works on aquatic systems and water is so important to human well-being and sustainability, he's also crossed that bridge between the social and natural sciences, and published a number of influential studies about the importance of water resources, equity, and more. So I won't go more than that, and please join me in welcoming David to E3B for what I think will prove to be a fascinating seminar. Thanks, David.
Well, thank you so much for that introduction. I'm really pleased to have met many of you virtually today, and also really happy that the rest of you that I didn't get to meet were able to join for this presentation. So my name is David Seekell, and I'm an ecologist currently based in northern Sweden. And the core of my research program here focuses on whole ecosystem experiments. And these are studies during which the entire ecosystem is perturbed to test a hypothesis at the scale that's most relevant to global change in ecosystem management. So these aren't controlled experiments, and there's not really a null hypothesis of no change like you might have in a small scale laboratory experiment. Rather, we really go into these experiments with good reasons to believe that our perturbation will cause a response. And what I'm mostly interested in is the magnitude of the response and evaluating if that matters among all the other ecological processes, or if it matters from a management perspective. And I feel that these types of experiments are really valuable because they're typically the only way to conduct tests of a known perturbation that includes both the full complexity of a natural biological community and the large-scale physical and geochemical processes that constrain and interact with those biological communities. Let's see. So my work specifically focuses on lakes, which are really advantageous for whole ecosystem experiments because most lakes are small and they have relatively discrete boundaries, and because the animals that we study in them can't run away from us. And so this is a picture of my current whole lake experiment. In the foreground, you can see an experimental lake where there's lots of equipment that's used both for perturbing the system and measuring the response. And then in the background, there's a reference lake. It's very similar, except there's no perturbation. And we monitor it to understand variations that might be caused by changes in atmospheric conditions or in the watershed. So what I want to do for this presentation is describe to you how I use whole lake experiments to address three basic questions in ecology. And the first is, can ecosystem tipping points be anticipated in advance? How does the transfer of organic material across ecosystem boundaries regulate populations and chemical cycling, particularly in the recipient system? And then how climate change impacts ecosystems. And each of these questions really requires large-scale studies to include the relevant processes. And each question, I think, has the potential to directly inform ecosystem management. So, tipping points. A tipping point occurs when some environmental change creates new feedbacks that fundamentally alter the character of an ecosystem. And tipping points often have severe consequences for human well-being through losses of ecosystem services. They typically occur without warning, and this is really problematic because they're often difficult or impossible to reverse. So I'll just give you an example of a tipping point, and that's eutrophication of lakes due to nutrient pollution. And what this is a tipping point that results in excessive plant production, harmful algae blooms, and fish kills. And it's a global problem, and it's likely to continue to intensify in the coming decades because of the increasing human population, demand for food, and conversion both for urban lands and for agriculture. And the economic losses attributed to this tipping point include the cost of water purification for human use, the losses of fish and wildlife production, and the loss of recreational amenities. And conservative estimates put these losses in the billions of dollars per year in the United States alone. So this tipping point happens because nutrient pollution creates new biogeochemical feedbacks within lakes that fundamentally alter the ecosystem state. So in most lakes, phosphorus is the growth limiting element for phytoplankton. When you start with a nice clear lake, when phosphorus enters the lake from the land, it settles in deep oxygenated bottom waters where it forms insoluble complexes of iron. And this prevents internal phosphorus cycling, which limits algal production. And that perpetuates a cycle of low phytoplankton biomass, also called oligotrophy. Now, when excessive masses of phosphorus begin to enter a lake, for example, from runoff from agricultural lands, transient phytoplankton blooms occur in the surface waters. When phosphorus loading exceeds a certain critical tipping point, the lake bottom waters become anoxic due to the oxygen consumption by phytoplankton decomposition. The phosphorus that was previously stored in the sediments becomes soluble again, and it starts diffusing back into the water column where it feeds further phytoplankton growth. This creates a new feedback that can maintain undesirable phytoplankton blooms called eutrophy. And it can be so strong, this feedback, that it will maintain itself even if the external phosphorus supplies are cut off. And what that means is that the tipping point is not easily reversible. And it would be much better if we could use adaptive management to avoid crossing the tipping point in the first place, rather than crossing it and trying to return later on. So I've worked for a number of years to develop methods to anticipate these types of tipping points. And with collaborators, I've identified unique early warning signals that appear in environmental monitoring data before tipping points. And so I'll explain the concept behind how these early warning indicators work, and then I'll explain to you how I use whole ecosystem experiments to do proof of concept tests. So this line is data simulated from a model of grazing by population that grows logistically. And there's two stable equilibria representing alternate stable states separated by an unstable equilibria that's a tipping point. And I've left out all the units and everything because the only thing that matters is the overall pattern. So the model was parameterized to be far from a tipping point, and then data was simulated, and an experimental perturbation was introduced into the model. And what I want to draw your attention to is that the value returns to the equilibrium state pretty rapidly after the perturbation. Now, here's some data simulated from the same model, but reparameterized to be close to a tipping point. And what I think is really clear here is that it takes much longer to return to the equilibrium after perturbation. And this observation is really the crux of the theory behind early warning indicators for tipping points. And that is, as an ecosystem approaches a tipping point, its recovery from perturbations becomes progressively slower. So this is known as critical slowing down, and it can be used to provide early warning of an impending tipping point. So a pretty robust literature shows that this slowing down process creates unique statistical signatures in ecological time series, including increased variance and shifts to low frequency variance. And that's a really good thing because it's not practical to go out and repeatedly perturb the world's ecosystems. It's not practical. It's probably not even legal in most places. And so these variance-based metrics are more easily measured and potentially practical for field application. So with collaborators, I've conducted a whole ecosystem experiment designed to test these variance-based indicators. And this type of test is really needed because real ecosystems, they comprise complex and higher order interactions and patterns of variability and measurement error that can all impact the ability to detect early warning indicators. But there are also things that are really difficult to replicate in more controlled settings. So what we did is we instrumented 2 forest lakes in the Upper Peninsula of Michigan, and then we experimentally manipulated the food web of one lake to cause a tipping point. The lakes were pretty similar in all of the physical and chemical characteristics. And biologically, the species composition and the food web structure were also basically the same. So this is kind of the upper part of the food web. There's piscivores, in this case, largemouth bass, which is a common temperate fish species, and they eat planktivores, the pumpkin seed sunfish. Those in turn eat zooplankton, which are diverse, but the important ones are really the Daphnias. Now, there's also juvenile predators which compete with the pumpkin seed, with the planktivores. And this creates like a food web structure that's known as a trophic triangle, which is relatively common in fish communities. And it's really cool because it can create positive feedbacks that drive either the predators or the prey to dominance. So how that works is that if you start with a lot of predators, they suppress the planktivores. And this facilitates the growth and recruitment of the juvenile piscivores into the adult piscivore population. And then they perpetuate their state of piscivore dominance. But what's really cool is if your initial condition is lots of prey fish, they outcompete the juvenile piscivores. And this creates a reproductive bottleneck for those fish. And it makes it so few of them grow and recruit into the adult piscivore population. So the adult piscivores are suppressed, so there's lots of planktivores, and the planktivores perpetuate a cycle where they're actually regulating the food web. So prior to our study, the reference lake was in the predator-dominated state, and the experimental lake was in the prey-dominated state. And they had been this way for at least 30 years, which is about three generations of the predators and about twice that for the planktivores. So what we did is over the course of 4 years, we slowly added adult piscivores to the prey-dominated experimental lake. And what this did is it broke the recruitment bottleneck, which caused a tipping point into predator dominance. Throughout the study, we gathered diverse physical and chemical and biological data at different spatial and temporal scales. But I'm only going to show you a small subset of that. And what I'm going to show you is called catch per unit effort, which is an index of relative fish abundance that we measure daily for the planktivores based on distributing minnow traps around the edge of the lake. So this is a picture of our study in action, the actual perturbation itself. And now what I want to do is walk you through some of the detailed results from the study. And what we have here is 4 summers worth of catch per unit effort data that's concatenated into one time series. And what these values are the mean number of fish caught in minnow traps each day. And these are the planktivores that are being caught. And those traps are around the outer edge of the lake. So in red are the catch per unit effort from the reference system. And these values were low and stable throughout the entire study. And it's very sensible that they were low because it was a predator-dominated state, so there's not a lot of planktivores around. But it's notable that there wasn't a lot of variability because this indicates that there weren't any real remarkable events happening in the watershed that may confound the study interpretation. So the vertical blue lines, the dates that we added adult largemouth bass piscivores to the experimental lake. And in black is the planktivore catch in the experimental lake. So prior to the first largemouth bass addition, the planktivores were really abundant based on the trap catches. And there was a lot of high frequency, that's day-to-day variability in the trap catch. Immediately after we added largemouth bass, the catch began to decline. And I think it's really clear that the variability shifted to lower frequencies, and meaning that you go from high day-to-day variability to longer-term oscillation. And this is exactly the type of shift in variance frequency that's caused by critical slowing down, and was expected based on mathematical models. By the time the last largemouth bass addition occurred, the daily catches were low and stable in the new predator-dominated state. So one of the things that really excites me as an ecologist about this project is that there are some complex dynamics specifically related to the oscillations, but we can explain all of these through real ecological mechanisms. They're not just some dimensionless mathematical model. So for example, these transitional oscillations are due to the interaction of fast and slow ecological processes. In this case, the piscivore abundance is a slow changing variable, something that's driven by our experimental additions and by the annual reproductive cycle of the species. Now, the planktivore catch is a fast changing variable, and there's large magnitude of intra-annual variability that's driven by their behavior, specifically their decisions to move between the foraging zone and refuge, as well as the decisions to shoal together in response to predation risk. So basically what I see here as an ecologist is that the first additional largemouth bass was too late for them to reproduce, but it was still early enough for them to begin suppressing the planktivores. As a consequence of this suppression, the largemouth bass had a really big year class during the second year, and the direct and indirect predation suppressed competition from the planktivores, such that a lot of these young of the year largemouth bass, they grew really rapidly. They survived at high rates through the winter into the third year. Now, these juvenile largemouth bass grew so fast that they were able to prey on the pumpkin seed sunfish midway through the third year. And that's the point when the recruitment bottleneck was broken and the tipping point occurred. And that's kind of marked in this gray line. Now, that third year, the adult largemouth bass pulled off another big year class, and the system was truly pushed out of its oscillatory phase and into the predator-dominated state. So this kind of extended delay between the annual reproductive cycles of largemouth bass allowed time for the cycles to form in planktivore dynamics, and that really allowed us to see the changes in variability before the tipping point. So here is a figure showing an early warning indicator calculated based on variance. And this specific indicator has a threshold that indicates when it's meaningful. So if you're in the red, it's a meaningful early warning indicator. And if you're in the green, there's no early warning. And it's just one of many different indicators that have been developed and tested. And this specific one, I'm going to draw your attention to that there's no early warning in the reference system through the entire 4 years. And there shouldn't be because there was no perturbation. But what I think is really clear is that there's early warning for basically the entire second year of the study and into the third year until the tipping point occurred. And then there was no early warning for the balance of the study. And this is, of course, because the tipping point already passed and there was nothing left to warn about. So this is really the first ecosystem scale proof of concept that early warning indicators can be detected in advance of a tipping point. Now, what's cool is that critical slowing down and its related early warning indicators, changes in variance, are really general phenomena and they're not restricted to ecosystems. So after our experiment, other research group identified early warning indicators before tipping points in a wide variety of complex systems, including in economic records before the subprime housing loan crisis back in like 2008, before the self-termination of epileptic seizures, and before shifts in social networking activity on Twitter. Even some medical doctors say that they found the same early warning indicators before the onset of clinical depression. So I think that's really cool. And I think that although my own research typically focuses on lakes, I think it's really exemplary of how fundamental ecological research can generate tools and concepts that provide diverse benefits to research and to society. So the next question I'm studying is with whole lake experiments, is how the transfer of organic material among ecosystems regulates populations and productivity. And lakes are really ideal systems for asking this question because they sit on depressions on the landscape. And that makes them really natural collecting points for organic material that's flushed in from terrestrial ecosystems. And most of this terrestrial carbon comes into lakes as dissolved organic carbon. And what this is an amalgam of thousands of different, really poorly characterized organic compounds that collectively serve to stain the water brown, like Coca-Cola brown or coffee brown almost when it's in high concentrations. So this terrestrial carbon, dissolved organic carbon, has a strong role in regulating lake ecology. So for example, lakes with high terrestrial carbon loads have less fish biomass compared to lakes with lower terrestrial carbon loading. And so for example, if you randomly selected pairs of boreal lakes in Sweden that have the same depth, there'd be a 77% chance that the lake with darker water will have less fish biomass than the lake with clearer water. And this is because fish in lakes with dark water have lower growth rates and lower lifetime fecundity. So terrestrial carbon concentrations are increasing in lake waters across the Northern Hemisphere. And this finding, along with the previous figure, collectively suggest that fish biomass will decrease in the future. And this is particularly problematic for regions where recreational and small-scale commercial fishing contributes significantly to local economies. So for example, where I live right now in Sweden, many communities which have the strongest impact of fishing on the local economy are also in regions that are most likely to be impacted by this change in watercolor. Now, there are two competing hypotheses in the literature that link terrestrial carbon to the more proximal impacts on fish biomass, the low growth rate and low fecundity. And the first hypothesis is that terrestrial carbon it stains the water brown, so that reduces light penetration, and should follow that it would limit photosynthesis and therefore secondary production in the ecosystem. This is called the resource limitation hypothesis. The second hypothesis is that terrestrial carbon reduces the depth of the oxygenated mixed layer. So most of these forest lakes are two layers of water. The top, which is exposed to the atmosphere, has oxygen in it, and the bottom layer is not exposed to the atmosphere and it has no oxygen in it. Lakes with darker waters typically have thinner oxygenated layers, and the habitat limitation hypothesis says that in the darker lakes, there's just not enough room to support large fish populations because of this shallow mixed layer. And to some extent, you could say, from a purely predictive point of view, it doesn't really matter which pathway really makes the connection, because they have the same basic outcome. You have less fish. But they actually have really different implications for management potential. And specifically, we know how to manage for the resource limitation hypothesis. We have good ideas about how to clarify lake water and how to do this at scale. But in terms of adding oxygen to lakes, it's very energy intensive and expensive, and it's something that can't be scaled. So it's something that isn't really effective. There's no effective management strategy. So I'm currently conducting a whole ecosystem experiment designed to test these two hypotheses. And the ecosystem scale is really necessary to capture the large-scale physical processes like thermal stratification, and as well as the diversity of habitats that are needed to have a realistic fish response. So the way that I'm approaching this right now is by directly testing the habitat limitation hypothesis. So what we've done is we've added oxygen to the anoxic bottom waters of a small forest lake. And we did this by building a special machine. Well, first we had to build a power source, and then we built a special machine that would add water in, or add oxygen in, without changing the water temperature and without changing the thermal stratification. And this was a really significant effort because it's difficult to find study lakes that don't have conflicting uses. So we had to go deep into the forest where there are no roads. You can see in this satellite image, there are no roads, there's no power source or anything like that. So we have to start from scratch in building our infrastructure, and then build our own custom equipment, both to produce oxygen and then get it into the bottom of the lake without disturbing temperature. So we've completed 3 full years of the study, 2 pre-treatment years and 1 treatment year, and we've successfully raised the oxygen concentration of the bottom waters to surface water levels. And we did this without changing water temperature or thermal stratification. So this is, I'm really proud of this effort. It's something that's very difficult to do. And to date, we haven't found any evidence that the fish use the additional oxygenated habitat. And in fact, there's so little evidence that we didn't find really any evidence they actually went there at all. I couldn't even make a graph to show you. There's no fish there. So it'd be just some axes with no data on them. And this suggests that habitat limitation isn't the primary mechanism limiting fish production in lakes subject to heavy terrestrial carbon loading. And that's actually a good thing, because it means that we have management options. So in the really immediate term, I've continued oxygenating through the winter and hope to do it within the next summer, as long as the university is open. And this could allow more time for potential changes. So maybe one season wasn't enough time for the various bugs that fish feed on to really colonize the habitat. We'll see. We're also analyzing high frequency telemetry data that we collected to understand if maybe the habitat was used for really short periods of times, for example, during heat waves as an important thermal refuge, but not used during the rest of the year. So maybe there was some importance we just missed. Beyond this summer, what I'm going to do is transition out of testing the habitat limitation hypothesis to more directly test the resource limitation hypothesis by experimentally clarifying the lake water. If that works really well, we'll be able to have a real intervention to go back to the government with, so that there's real options to manage fish populations in response to this type of environmental change. So this third question on how does climate change impact ecosystem function is a big one. I think it's of clear immediacy and importance, but it's also of great difficulty, and in part because of the scale of the issue, in part because of the diversity of Earth's ecosystems, and in part because of the diversity of ways in which humans are part of ecosystems or otherwise interact with ecosystems. So I address this question through the study of Arctic lakes. And lakes in the Arctic, at least in the Swedish Arctic, have always been used by humans, and historically supporting the traditional lifestyle of indigenous people, in this case, the reindeer herders. Today, they're primarily used for fishing tourism and small-scale commercial fishing, particularly focused on Arctic char, which is a culturally and economically valuable species. Now, these industries are important to the local economies in northern and mountain communities, and the Swedish government actually promotes exploiting this resource as a means of economic development in rural communities, specifically the ones where traditional industries like mining are employing fewer people, and that's due to mechanization and now automation. And historically, the lakes in the Swedish Arctic have been really good char habitat. That's because they're really pristine and they mix regularly. And this means that they have the same cold temperature and high oxygen concentration throughout the entire lake. And this is really important. This species is really, really sensitive to temperature and really, really sensitive to low oxygen concentrations. Now, in the past, there's sometimes been transient periods of thermal stratification. That's when the surface waters become too warm, but it's not really a big deal because the fish can just move deeper into the deeper oxygenated layers. However, recently, we observed some really prolonged periods of thermal stratification, up to 120 days, which is pretty much the entire growing season. And with this stratification came fairly rapid oxygen depletion in the lake bottom waters, which was surprising to us because this is usually something you see in very dark lakes or in very polluted lakes. When this happens, the Arctic char habitat rapidly compresses because the surface waters are too warm, but the bottom waters don't have enough oxygen. So you can go from having a whole lake's worth of habitat to having almost nothing in the course of a few weeks. And climate change should make prolonged periods of stratification more common through a number of different mechanisms that all go in the same direction. Rising air temperatures, in some regions declining wind speeds, these can induce stratification, but also the catchment vegetation development, which includes tree line advance that creates wind sheltering. And it also, of course, this vegetation fixes carbon, which ends up in the soil and during the snowmelt in the spring flushes into the lakes. And that carbon is colored, which absorbs light and warms the waters, helping to stratify them. So there's a whole bunch of different mechanisms. And through our monitoring data, we've been able to construct simple statistical models that predict that over a third of mountain Arctic lakes in Sweden will experience this type of rapid habitat compression within 17 to 50 years. And that's only considering the sheltering effect of the advancing tree line. So it's an alarming observation to have, and it suggests some major challenges for the fish populations and the rural communities that have economies based on them. So relative to the fish more specifically, early monitoring revealed that char populations were really decimated after a recent summer of prolonged stratification. So here on the left is a basic pattern that we observed in 2018, which was a summer that had widespread thermal stratification. And the longer the stratification, the less fish that we caught in the lakes. And on the right, you can see for one specific lake, a study lake, almost 50% reduction in catch, comparing the summer before stratification was widespread and the summer after. Unfortunately, we didn't have the summer the stratification actually happens because our permits for this lake only permit biannual sampling. And because we weren't expecting stratification to be an issue, we weren't set up to measure for it. However, we know that stratification was widespread, and based on long-term records from other systems that are measured annually, we know that a drop like this is historically very large compared to what's happened in the past. And these types of observations and more are very suggestive, but they don't really put us in a position to link stratification and habitat compression to these actual changes in fish population, and they don't get us where we need to be to understand what future conditions will be like. So right now, what I'm setting up to do is to experimentally stratify an Arctic lake to identify where the oxygen is going in the bottom of the lake. So we don't actually know because our own measurements indicate that these lakes produce more oxygen than they consume through processes like respiration. And most of this oxygen production comes from benthic algae. So it was a surprise to see that the oxygen was disappearing. So we'll experimentally stratify a lake and to identify the mechanisms behind the oxygen loss and what the implications are for fish physiology and their reproduction and population dynamics, and eventually what that means for the long-term trajectory of the region. So the way that I plan to do this is by erecting special wind conditioning fence around the experimental lake. And what this is designed to do is both to reduce the wind speed and to reduce the amount of turbulence. And the idea is to really shelter the effect that's created by the advancing tree line. Then we can monitor oxygen budgets and fish populations, among other things, to answer a variety of questions. And those are, why does oxygen deplete in the bottom of these lakes? What are the impacts on physiology, and how does this translate to population dynamics? What lakes will be impacted, and what is the time frame of impact? And this is something that's addressed with a combination of these experimental results, models of lake physics that can be applied across the landscape to predict future conditions, and also the results of comparative analyses. I'm also interested in contributing to the evaluation of potential impacts on fishing and tourism industry. And we can do this even in very basic ways. So for example, if our models and experiments predict that only very remote lakes will be impacted, it probably won't impact the local economies of the fishing communities that much. However, if all the lakes around the fishing communities are going to get slammed and it's going to happen really soon, then it could be pretty bad. Finally, I'm interested in exploring connections with these fish problems to issues of equity and social justice. So right now, the lakes are currently owned by the government, but the courts have ruled that they need to be returned to the indigenous people. That's the Sami reindeer herding communities. And it hasn't gone without notice to us that these lakes were kind of, acquired hundreds of years ago, kept for some time, and then returned just as the most valuable part of them is about, potentially about to collapse. And so we made some connections with researchers studying indigenous issues, and we'll also kind of look at the ecological processes through those lenses. So I just want to take a few minutes now to describe how I see my work fitting in at Columbia before taking any questions about my research. So first and foremost, I think that my interests both complement and extend the scope of teaching and research that's already occurring within the department. So there's a lot of, I think, geographical and maybe topical overlap in terms of foundational concepts between my own research and some of the research already happening in the department, especially related to the Arctic, biogeochemistry, ecosystems, and then food and sustainability to some extent. Some of the areas that I think that my work might extend the scope of the department's focus includes on my focus on aquatic ecosystems, especially studies related to aquatic organisms such as fish population dynamics and ecology. I also think that my work fits well within the broader university, particularly within the Earth Institute. Um, the Earth Institute's scope is so broad, but in general, the topical themes covering ecosystems, water, and food intersect really well with my research program. And there's even things like Columbia Water Center and the Hudson River Field Station that, that align really well with my research program. Finally, New York is really an ideal location to have a research program in aquatic science and aquatic ecology and to integrate teaching in with research. And this is in part because there's great connectivity from New York City to Arctic and boreal field sites, but also because there's really an outstanding diversity of aquatic ecosystems in pretty close proximity to maintain a local research program. That's particularly important for integrating teaching with the research. In particular, some of my future research would be based in the Adirondack Park and on the Hudson River. And I would even try to establish a small program based on the lakes and reservoirs at the Black Rock Forest. And this would be particularly convenient for trying to tie in teaching with the research. And one reason why this is really possible to do, and possible to do relatively quickly, is because there's absolutely fabulous existing data that exists for New York's aquatic ecosystems. There's so much known that you can dive right into asking tons of really interesting questions instead of getting the background information. And I think that it's relatively unique in that way. So with that, I want to thank you again for joining me for this presentation. I'm really happy to take any questions you have about my research, especially the lake experiments, and yeah, any questions you have. Thank you.
All right, so we've done this before by using the raise hands function. So if you go to the participants list, you'll see that there's a function at the bottom for raising your hand. And then I'll keep track of who would like to ask a question. While you people are raising your hands, I will start off by asking a question, if I could. Particularly interested in the tipping point question. My perception was always that if you have a system that is experiencing chronic change, that you may not see changes in certain measures for quite some time, and then all of a sudden it flips. But I also understand that if there's both chronic change and a sort of a cycling or a sinusoidal process, like seasonality or going from extremes in temperature or pH or things like that, that doubles the possibility that you can see a regime shift. In these lakes here, like with the phosphorus system, it seems like this is focusing a lot on the sort of, and maybe I have this wrong, on the constant pressure, the chronic change. What about the changes that we're seeing that are occurring in the sort of cycles, in the amplitudes of those cycles as climate changes and as other anthropogenic forces are changing?
So in principle, some of the same methods, early warning indicators, will work, but oftentimes it depends on measures of the size of the cycle that are really difficult to capture in practice, but can be done in theoretical models. So so in principle we can do. In practice, I don't know of any proof of concept on that type of system. I would say that we've proved a concept in a very we picked the system where we had the best opportunity to prove the concept. And what we haven't done is generalized that it will work much beyond that. We've really just proved the concept. And so there's a lot of potential for exploring how these methods may or may not work in systems with the types of dynamics that you describe.
All right. I see that Ruth, Ruth has her hand up.
Yeah, okay, thank you for the presentation. My question is along the same lines that Shahid asked and you were just discussing. So in looking at tipping points and criticality, fundamental concepts in systems, you mentioned that you study this in closed lakes because that's where it's possible to to study these systems. So how to move beyond that single lake system? Steve Carpenter also looked at these questions in lakes. So how do you find another system where you can test these concepts?
So in principle, this will work in some terrestrial ecosystems, especially, for example, in dryland systems before desertification, which is a particularly important tipping point because about 2 billion people live in drylands globally, and a lot of them are the most impoverished people on Earth with the lowest levels of food security. So it's a very important tipping point, and you can see changes in variability in the patterning of vegetation that happen in a predictable way before the before desertification. That said, this is it can be really difficult to apply out into other ecosystems, especially some terrestrial systems, because, or at least to do as proof of concept, because kind of the characteristic scale of variability and characteristic spatial scales can be larger and more difficult just as a logistical thing to collect the necessary data for. So there's been some proof of concept that shows the early warning indicators show up in dryland systems before desertification, but actually making the jump between proof of concept in the best case scenario to actually rolled out for real environmental management, that's a bridge that hasn't been crossed yet. The proof of concept has really been in the best case scenarios, and there's a lot of disconnects between the ecological theory and these, best case scenario proof of concept studies, and then actually what we need to be able to do in practice. And a lot of it has to do with, describe it. We started with theory, and it works really well. It's really robust mathematically, and but there are sometimes assumptions like you have 10,000 equally spaced data points with no missing data and no measurement error. And obviously that becomes a problem when you have to collect data in practice. And so kind of dealing with the assumptions of the ecological theory and reconciling that with the practicalities of actually collecting data, especially in some systems that are a little bit more difficult to collect data in than a small lake. This is really the biggest challenge in this type of studies.
Oh, I see Duncan has his hand up.
First of all, really nice talk. This is cool stuff to see. My question is also about early warning indicators of regime shift. So the theoreticians have come up with a bunch of different ways that people can detect regime shift to look for various things. So and you talked about one of them that you used in this study. I'm wondering if you can give us a quick overview of the different types of things people look for and why you chose the method you did.
So in general, the methods fall into two classes, and this is a broad generalization. And one set of methods falls within tracking changes in autocorrelation over time, the similarity of your measurements between time steps. And in general, as a system approaches a tipping point, that autocorrelation spikes. up to be near 1. And what happens when this happens is that your high frequency variance goes down and the system enters into more oscillatory phases as you kind of get up there. The second class of indicators relates to variance, specifically increases in variance and shifts to low frequency variance. And the indicator that I showed you is actually kind of a hybrid between these. It's actually a measure of autocorrelation of variance. And the reason why I showed that is because it's really well, honestly, from an online presentation where there's not a lot of time to get into the, all the details, it seemed like it'd be the most obvious to make the point. It's also because it was in my dissertation, and so I have a preference to show my own work. And finally, because the variance-based methods are more robust than autocorrelation-based methods and are less impacted by choices of sampling frequency than autocorrelation-based methods. So this is kind of the confluence of several different things that led me to present that data.
All right, so Kevin has his hand up.
Hi, David. Thanks for a really nice talk, or at least the part that I caught. And I want to apologize because I came in very late, so I may be asking a rather naive question. But with your Arctic lakes, you said that you have a bit of a mystery about oxygen levels and perhaps under accounting for or missing oxygen. And my question is twofold. One, is this a permafrost system? Is the lake actually on? Is there permafrost under the lake? And second of all, have you guys thought about photodegradation and loss evasion of CO2 to the atmosphere from the aquatic system?
So it's there's for those unfamiliar with the north of Sweden, we have discontinuous to sporadic permafrost. Which means it's there, but it's not like a complete coverage like you might find in parts of Siberia or in parts of Canada or Alaska. And a lot of these lakes don't really have permafrost around them, but I mean, there could be that we don't know about because not all of them fall within the areas where there's really high resolution permafrost mapping. At least to my knowledge, I haven't seen the maps and that extend to all of our systems. So it could be there and we don't know about it. And certainly the melting of the permafrost transfers organic material into lakes, which can be mineralized by microbes to consume oxygen. So that's one way it could go. Another way it could go is if the water is somehow offset by groundwater inputs of groundwater of depleted oxygen. Another option is that the oxygen can be consumed through chemical processes or photo processes. And so, for instance, there's been studies in Alaska suggesting that these are really dramatic processes that create tremendous amounts of carbon dioxide, which is emitted from the lakes. In Sweden, based on measurements and modeling, we don't think that photo production of CO2, consumption of oxygen, is nearly happening at nearly the rate suggested, like in the science paper that came from Tulik a couple of years ago. We see much lower than that. But certainly it's something that's happening, and it could be the answer. We're not really sure because we were actually doing a different study and happened to catch all of this on accident. As far as any of us have known, they've always been known to be well-mixed systems, cold and with good oxygen. So we were really caught off guard by the stratification and the fact that the oxygen would draw down and draw down very rapidly. So I don't have all the answers yet, but we would, when we do the experiment, so we're starting to do the preliminary data collection this summer, and when we get the full thing running, we're really going to pull through all the different alternatives and try to get to the bottom of it.
Thanks, okay. So Sarah has her hand up.
Hi, yeah, thank you for a cool talk. I again am going to ask a question about the experimental lake setup that you talked about at the beginning with the two lakes where one was kind of this dominated by the largemouth bass and the other one by the pumpkin seed carnivory of the largemouth bass. Um, and I'm wondering, let's say that you added largemouth bass into that one lake and you saw this regime shift, and I'm wondering what would happen if you went back in and preferentially fished out the largemouth bass again, what you might expect to see if you think you might see, again, a shift back to the state where it was before.
I would love to do that study, but we haven't pursued it because we're not confident we could actually get it funded. It's really hard to get funding to replicate or do similar studies once a whole ecosystem experiment's been done. There are some exceptions, but in general, it's very difficult. In principle, you should be able to fish it back past a tipping point, and that would lead to planktivore dominance again. And however, this actually depends on what the relationship is between fish abundance and the number of fish you catch when you go fishing. So like what the functional response curve is. If the fisher was the predator and the fisher the prey, and you can have like different functional response curves in ecology. The same thing kind of works to some extent in fishing, like recreational fishing. And it should be, with the behavior of most freshwater fish, they have what's called hyperstability, and that means that the catches remain high until right before the population collapses, and that means that you should be able to quite easily fish the largemouth bass past a tipping point. There are some species that actually, this kind of functional response of fishers is actually different. It's called hyperdepletion, and that's where just a little bit change in population abundance causes a really rapid decline in catch. In those types of species, it would be really, really difficult to fish the system backwards. But those types of species are pretty rare. There's, probably only a handful of them. Largemouth bass, we could definitely do it. What I think is more likely to happen in this situation, more likely than us getting funding, is that the largemouth bass population will continue to grow, and it has since that study finished. But it's going to break into some oscillatory dynamics related to cannibalism. And because these fish eventually, when the population is large enough, they'll develop into a stunted adult population and they'll begin to cannibalize their young. And this will cause some population cycles, probably on a period of 8 to 10 years or something. Eventually, what's going to happen is that there's going to be a really hard winter that occurs at a low point in that cycle, and that will kill off enough fish to flip it back, maybe. At least that's my guess. Only time will tell, but I suspect that almost kind of like there's like this famous ecological theory called the paradox of the plankton, where enrichment increases the chance of extinction due to the creation of oscillatory dynamics. That's kind of about what I would expect to happen here, but only time will tell if it happens naturally like that.
Oh, thank you.
All right, so Matt has his hand up. Matt?
Hey, David, thanks for the talk. A lot of interesting stuff. You showed some data about Adirondack lakes shifting toward a browner regime, and I think you mentioned it was happening in Sweden as well. And you talked about the consequences of that. But I'm not sure if you addressed the causes. Why are lakes getting browner?
Yeah, I didn't address the causes, and that's because I get carried away and talk too long about it. There's a variety of causes, which include climate warming, shifting patterns of precipitation and snow, the shifting chemistry. Actually, recovery from acid rain can promote browning like this. Permafrost melt in northern regions, and the development of vegetation and catchments. Changes in wind patterns in coastal regions and the deposition of sea salts into lakes. There's really a wide variety of things happening at different timescales and in different places that kind of all collectively create this kind of hemispherical pattern of browning. So it's a variety of different factors at different scales, all kind of going in the same direction. And certainly, I think in that figure it showed that not all lakes are getting browner. In the case of the Adirondacks, some are actually getting clearer, or some of them are having very little change. It's just in general, most of them are getting browner, and that's a pattern seen in by independent groups in many different regions.
Thank you.
All right, so Duncan has his hand up again.
I do have my hand up again. I wanted to follow up on your answer to Sarah's question, where you were talking about the paradox of enrichment in the bass-dominated lakes. If I remember correctly, the reference lake you had was a bass-dominated lake, and I think you said you had a pretty long-term observational data set on that. So what has happened in that bass-dominated lake before the time zero on the graph you showed?
So that bass population is cyclical on a period of about 8 years. That's related to cannibalism. And the adult bass population is stunted, also related to their really extreme dominance. That happens when they become really abundant, actually. So like if you wanted to manage that lake to get trophy fish for good fishing, you would actually fish out a bunch of fish. Now, the data that I showed you was data for planktivores. And they're not having that same cyclical pattern, in part because even on the down years of the cycle, the predation pressure is still extreme. So that's why the planktivore data was really low and constant throughout the entire 4 years. If we were to look at a longer, say 30-year record for the adult predator population, you would see really clear cycles related to cannibalism.
So in that case, you would predict cycles in the bass population, but not reverting back to the planktivore dominance.
I mean, I would definitely predict the cycles will break out in the bass population. They could, so it won't revert back on its own, I don't think, but it could revert back if there's some extreme environmental event. And one thing that does happen in northern lakes are sometimes there are extreme winter kills where, for whatever reason, the confluence of many events makes it so that most of the fish die off over the course of the winter. And it could be that one of these events is what does in the largemouth bass to flip it back. It could be that I'm wrong and it just persists in this state until someone comes in and does another experiment. Really only time will tell, I think.
All right, looks like there's we're over the hour, and it looks like there's no more questions. So I think it remains for us to thank David. I wanted to say that at this point, we can't do it virtually. We would all sort of retire to the lobby, and we would have cheese and wine and beer, and then we take it out to dinner to a nice restaurant, and we continue the conversation both informally and formally. But we'll have to do that in our mind, and hope to be able to do that at some other time. So I'd like to say to all of you, thanks for participating. Stay safe and stay healthy. And let's thank David for spending the whole day with us virtually one more time. Thanks, David.
Thanks, David.
And for staying up so late at night.
Thanks, everybody. It was really a joy to meet so many of you.
All right.
Thank you.
We'll be in touch with you. And by the way, we're going to set it up so that if people have more questions, they can continue asking questions electronically. All right? Okay. Bye-bye, everyone. And take care.
Bye.